So welcome everybody to today's webinar where we will discuss about overcoming challenges in vaccine development with my colleague Quentin Canel. So Quentin studied bioengineering at the Free University in Brussels, and there he also did his PhD in collaboration with GSK. Actually, the site in Belgium that is working on a vaccine. So Quentin already has an experience in the vaccine field. So we are really looking forward to hearing more about vaccine, Quentin. Thank you.
Okay, thank you, Francesca, for this kind introduction, and thank you to you guys for attending this short webinar about overcoming challenges in vaccines development. So to start this presentation, I would like to start by what is a vaccine and why it's important for the health system. Vaccines are products which produce immunity against a certain pathogen. It will do so by teaching our immune system how to react in case it will face a specific pathogen. So it's very important for different reasons. The first one is very obvious. It's because the vaccines will protect the patient against a pathogen and different dangerous disease. And it will protect us from this disease without having us to suffer from the disease. The next reason why vaccines are important, it's because maybe some people are in good health and they will be able to react against a specific disease, but it's not the case of all the population. And some specific population, like the elderly or the children, might be more vulnerable because their immunity is decreasing or their immune repertoire is not finalized yet. So these people can be protected with vaccination as well. And as we can see now, vaccines has a key role to play in the prevention of the epidemic and also to control it. Because maybe it will also protect us, but it will also help to prevent the transmission of the disease from one people to another. And this will allow us to decrease the exponential progression of specific viruses. And last but not least, vaccines has been found to prevent an estimated 2 to 3 million deaths worldwide every year. And vaccines are also acknowledged to be one of the way to completely eradicate several diseases, like it was the case for the smallpox. So now that we know why vaccines are important, let's discuss of what vaccines are made of. Vaccines will typically contain two different components, the adjuvant and the antigen. The antigen might be of different source. It can be either a live attenuated vaccines. So these vaccines will contain the virus or the pathogen which is live attenuated. So it's still living, but it's not able to trigger any disease anymore. Then we have the inactivated vaccine. So in this case, the pathogen is not living anymore, it has been activated. It is the case of the hepatitis A, for instance. Then we have what we call the toxoid vaccines, in the case where the disease is due to not the pathogen or the virus itself, but by one of the secretion of the bacteria, for instance. So, for instance, the tetanus is caused by a toxin, and in this case, we'll produce a vaccine that will have a toxoid, which is a toxin-like protein, but it's not harmful for the patient, and it will also, and one of the key benefits is that this toxoid will trigger immune response also against the toxin. Then we have what we call the subunit vaccine. Instead of injecting the entire virus or bacteria, so the entire pathogen, we'll only and specifically use antigen that is only one part of this virus, for instance. And it could be one of the proteins that allow the virus to ensure the host cell. And by specifically targeting this part, we will also impeach the contamination with this pathogen. Then we have what we call the conjugate vaccines. These vaccines are made of usually polysaccharides that are less immunogenic, and as they are less immunogenic, there is a need to conjugate them with a specific protein. And then when we have what we call the adjuvant, which is something that is not really specific to the pathogen, and the adjuvant will help to increase the immune response. Okay, so now that we discussed what are the vaccines and how they are made, let's go to the vaccines development. So the vaccines development process is a four-step process that will start with the exploratory phase. In this first phase, we'll try to understand what is the mode of action of the pathogen and what antigen could be selected in order to prevent the infection. So it will therefore mainly focus on the antigen identification and characterization, but also on how to formulate the vaccine, as well as which adjuvant could be helpful to trigger immune response to get rid of this infection. Then we will move to what we call the clinical and preclinical development step. So once we have a good vaccine candidate, we'll move to the in vitro test, to the in vivo assay, and start with preclinical testing, where we start experiments on animal to determine the safety and also the efficacy of the vaccines. Then once we prove that it was safe on the animal, we will move on human and on clinical studies, where we will still try to prove the efficacy of the vaccine on a higher number of persons. And once the non-toxicity and the efficacy of the vaccine has been proved, all the results will be reviewed by the regulatory agencies to get the approval to put the vaccines on the market. So this step is really key in the way that that we will inject a product, so a vaccine, to healthy patients. So we really need to be sure that it's not toxic and won't have any side effect that could be damages for the patient. And then finally, once it is approved and we can have it on the market, the vaccines producer will have to manufacture it at larger scale and with the high standard quality control for this specific product. So during this vaccines development process, there are two different technologies from nanotemper that could definitely help to solve different questions and different complex issue. So one of them is the microscale thermophoresis, which will allow us to measure the interaction between two binding partners. And the other one is the nano DSF that will allow to look at the stability of the protein, which is stability of the protein like antigen, which are very useful in the vaccines development. So to highlight the use of these two different instrument technology, I would like to share with you some example. The first one is about the measurement of the stability of the antigen structure during the vaccines development. For some vaccination, the structure of the antigen is really key, and that's the case of the dengue and the Zika virus. For these vaccines, the antigen is actually one protein that is present at the surface of the envelope of the virus. And to be highly immunogenic and trigger a high immune response, this protein need to be in a dimeric state. If it's not in a dimeric state, it won't trigger a high immune response. So this dimeric state is actually highly dependent on the concentration. And what you can see on this graph, which is thermal unfolding of the antigen at different concentration using nano DSF, you can see that the profile is very different according to the antigen concentration. You can see that at high concentration, you have only one peak. But at lower concentration, you have another shoulder here, and this shoulder is actually a decay of the dimeric state into monomer. So the higher concentration will definitely stabilize the antigen as dimer, meaning that we will only have one peak because the dimer will fall apart as monomer at the same time. Whereas it won't be the case at lower concentration, where it will first decay, the dimer will decay in monomer before. So definitely nano DSF was a key tool to determine if the antigen is still as dimeric state, and this measurement is key along all the vaccines development process. It could be for formulation, but also for long-term storage and for patient immunization. As you can guess, the dengue and Zika virus are vaccines that are necessary in countries where the temperature and the climate are rather can be at high temperature. The second example I would like to share with you is the characterization of the interaction between the antigen and an adjuvant. So one of the goals in the vaccine, sometimes we will get an immune response which is not high enough. And there is a need to increase this immune response by using adjuvant that will increase the immune response of the vaccines. And there are some adjuvant, and it is the case of this specific peptide. So this peptide as adjuvant will interact with the antigen. In this case, it's the ovalbumin antigen. And by interacting with this antigen, it will create a hydrogel structure. And this hydrogel will capture the antigen and will release it more slowly. And the fact of releasing the antigen slowly, it will be presented more and more to the immune system, and the immune system will trigger a higher immune response. And as we can see here, we were able to measure an specific interaction of this peptide for this antigen, with an affinity around 80 micromolar. But if you take another compound that is very similar to this one, but not able to create hydrogel, you can see that there is no interaction at all. And if you look at the release assay, you can see that for the one creating a hydrogel, you have only 60% that are released after 24 hours, whereas for the other compounds, which is not creating hydrogel, we have 100% that are almost released after 16 hours. So MST technology is a key technology for the measurement of in complex matrices like hydrogel in this case, but also in serum, for example.
Quentin, I have a question. So you wrote there and you mentioned serum. What can you actually do with serum? Which type of experiment?
Yeah, so I don't know if you remember, but during the vaccines process, we were speaking about the preclinical and the clinical stage. And in this stage, what we will have to do is to assess whether a vaccine is the efficacy of the vaccine. And one way to assess the efficacy of the vaccine is to look at the antibodies that are generated during the vaccination. And these antibodies will be generated, of course, in serum. And the serum is very complex matrices that contains around 100 milligram per ml of protein. So it's very important to have a technology that is not sensitive to high background of protein, that is very specific, and that could also work, that is independent of the different buffer we will work in, which is the case of microscale thermophoresis. Okay, so now as other example, I would like to speak about the measurement of the antigen stability in presence of complex adjuvant like aluminum salt. So once again, in this case, we have an antigen that need to trigger high immune response. But in order to produce this high immune response, this specific antigen in the field of HIV-1 need to be really in a specific conformation, which is a native-like trimer. And as you can see here, if it is in a native-like trimer form, it will generate high neutralization titers, which is not the case if it is not. And if we look at this antigen in presence of different adjuvant, which is the case here, when we look at the thermal unfolding of this antigen either alone or in combination with alhydrogel as adjuvant or with aluminum sulfate as adjuvant, we can see different profiles. So in the alhydrogel, we can see that it is quite similar as the antigen alone, meaning that it's still in the good trimeric conformation. But in the aluminum sulfate, we can see a slightly different profile with different peak here, suggesting that actually the trimer is falling apart in a mixture of monomer, dimer, and trimer. So the nano-DSF technology will allow the measurement of stability of complex antigen like trimer in presence of challenging adjuvants, which are aluminum salt, because aluminum salt will create some opacity of the solution when they are in mixture, which make them very difficult to analyze with different technology like PAGE or microscopy mostly. Okay, so that was it for the presentation. So I hope that During this short presentation, you show that nano-DSF and microscale thermophoresis are useful in different phases of the vaccine's development, like in the exploratory phase, where it can help for the antigen identification or characterization, as well as for the formulation and the antigen design. It could also be helpful in the clinical and preclinical development, where it can help to measure in complex matrices like serum, and also during the manufacturing and quality control step, for example, for batch-to-batch comparison, as we are working with biological material, is really key to assess whether one batch is similar to another.
So Quentin, regarding the exploratory phase where, you can work on antigen identification, characterization, you also talk a lot during the talk about infectious diseases and viruses. Mm-hmm. Can we, with microscale and thermophoresis and nano-DSF, study entire viruses?
Okay, so it's actually one of the key thing about our technology. So MST works with, is not only based on modification of the size, and we have been able to measure interaction with complete influenza virus, which is one of the key target in vaccines development. So MST has been shown to work with complete virus. And also for nano-DSF, there are some current applications that are performed with adenovirus vector, which are also complete viral particle, as well as VLP, which are also well used in the vaccine field. So yes, both technology are definitely useful for complete viruses as well.
Okay, that's great. Now, just a curiosity, is anybody already using microscale thermophoresis or nano-DSF to study COVID-19?
Yeah, so actually now the studies about COVID-19, there is a lot of interest, of course, for this really hot topic that is very important at the moment. And there are already different groups that are using MST and nano-DSF in the field of vaccines discovery for COVID-19, but also in the field of therapeutics to also develop different therapeutics against the COVID-19. So yes.
Okay, so now we are at the end of our webinar. I would like to thank everybody for watching it and participating, and also thank you, Quentin, for your presentation. If anybody has any question or is curious to hear more about microscale thermophoresis and nano-DSF, please check Nanotemper Technology website and contact us. And last but not least, we are supporting the global message of stay home and stay safe. Remember, we still don't have a vaccine for COVID-19. So thank you again, everybody, and goodbye.
Thank you.